Let's walk through 5-base DNA methylation analysis. In this study, I have already added 6 samples to the study. I am going to go ahead and create a new analysis by clicking the New Analysis button. I'm going to give the analysis a name, 5-base demo. Choose my analysis type as custom 5-base methylation, and because I have 6 samples here, I'm going to select all 5 base samples and run analysis. The analysis status has moved from in progress to complete. Let's go ahead and open this analysis. We see an ongoing import cohort task. Wait for the data import to complete. After the data is imported, hover over the 5-base methylation data node. Shows we have 6 samples and about 28 million features. Features refer to CpG sites. Click on Metadata tab. It has a sample table that consists of 6 samples. Click Manage under the Sample Attribute section. Then click Add New Attribute button to create an attribute called Sample Group. Under Sample Group, type in a value in New Category text box, then click the plus button to add category values. Go back to the Metadata tab. Click Assign Values and use the drop-down to select a value for the sample group attribute at each sample. Then, click Apply Changes to save the assigned values. If you have many sample attributes or have large number of samples, you can upload a sample metadata file to the study to automatically link sample attributes to an analysis. With this dataset, we ask 2 questions. The first question is which genomic regions show differences in DNA methylation patterns across the 2 sample groups? The second question, what are potential pathways involved for biological context? To start analysis, go back to the Analyses tab. Click to select the 5-base methylation data node. Go to Context-Sensitive Toolbox here on the right. Under QA/QC section, click 5-base methylation QC task to generate QC report. The 5-base methylation QC task has completed. We can single-click and open the task report here in the toolbox to open this output, or we can double-click to open this output and view the results. This QC report consists of plots and tables organized in 2 sheets. We are now looking at the metrics data sheet. In these plots, each data point is a sample. They are randomly spread out on x-axis. The QC metric is represented by y-axis. Each plot is overlaid with a violin plot to show distribution of the QC metrics. Plots in the top panel show methylation QC metrics, such as percentages of CpG methylation in samples, and conversion efficiency in the controls. Plots in the middle panel show reads QC metrics for sequencing and alignment quality. Table at the bottom is text representations of the QC metrics plots. Switch to M-bias sheet. M-bias plots are used to examine methylation bias across positions on reads. Generally, we expect M-bias to be consistent across all positions. It is common for the first or last few bases to have uneven methylation due to sequencing artifacts. Go back to the analysis pipeline by clicking the analysis name on the top of the user interface. After review the data quality, we are going to perform dimension reduction using PCA to explore source of variations among the samples. We specify parameter to use the top 100,000 CpG sites with the highest variance for principal components calculation. Keep the remaining settings as default and click Finish to start running PCA. After the PCA task is completed, double-click on the PCA node to open output. On the left, we see a 3-dimensional PCA plot. It can be rotated and viewed from different angles. To reset the plot orientation, click Plot on the left Tools menu, select Reset View. Each data point is a sample, colored by sample group. Here we see samples from Group A and Group B are clearly separated on the first PC. At top right, we have a scree plot that shows variants represented by each PC. Bottom right is a component loading table displaying correlation between CpG sites and PC. Click on the analysis name to go back to the analysis pipeline. From the PCA, we see there are differences in methylation profiles between the two sample groups. Next, we are going to use statistical tests to identify genomic regions that are differentially methylated across the two sample groups. Select DSS as method to use for differential methylation. Click Next. Select sample group as factor to analyze. Click Next. I want to compare sample group A versus sample group B. So I drag and drop A to numerator box, B to denominator box, then click Add Comparison. The comparison is now added. Click Finish to start running DSS. The DSS task generates two outputs, one at loci level, differential methylated loci, or DML, another at region level, differential methylated regions, or DMR. We are going to focus our results interpretation and downstream analysis on DMRs. Double-click on the DMR node to open the differential test report. Each row in the report table is a region. On the right, there are test statistics for comparison A versus B. The results are sorted by absolute value of area stat statistics. The larger the area stat, the stronger evidence of differential methylation. Click on volcano icon next to the comparison name to visualize the differential test result. On the plot, methylation difference is represented on the x-axis. Absolute area stat is represented on the y-axis. We are going to use the style dialog to color the regions in this plot by significance, and use statistics dialog to define significant thresholds. Here, we set methylation difference at -0.2 and 0.2. No cutoff for the absolute area stat statistics. Now, the hypomethylated regions are colored in blue. The hypermethylated regions are colored in red. The plot can be exported to local machine by clicking Export button on top, and choose the preferred format, size, and resolution. Click on the analysis name to go back to the analysis pipeline. Next, we are going to filter the DMRs using differential analysis filter. Select metadata as filter type, then specify filter criteria to include regions with methylation differences greater than 0.2. Click Finish to generate a filtered node. From the filtered DMRs list, use Annotate Regions task to annotate the regions with gene name. Gene name is required as input for downstream pathway analysis. Select an annotation model. In our case today, this is Human Data HG38 for the genome assembly. And for the annotation model, we're going to use the GenCode Genes Release 44. Use the default settings for the rest, and click Finish. Double-click on the annotated regions. Node opens the task report. On the top, there is a pie chart showing gene section breakdown among the regions. At the bottom is a table where each row is a region, and columns are genomics annotation. Use optional columns to show, or hide columns display on the table. Click on the analysis name to go back to the analysis pipeline. With regions annotated with gene name, we are ready to find potential pathways involved for biological context. Select the Annotated Regions node, then choose Gene Set Enrichment task from the context-sensitive menu on the right. We support two types of databases for enrichment: KEGG Pathway Enrichment or Gene Ontology Enrichment. In this demo, we're going to use the KEGG Pathway database for human. Select Gene Name as Feature Identifier, then click Finish. Our KEGG Pathway Enrichment has completed. Open this task report. In this report table, each row is a pathway, sorted by significance of the enrichment. We can filter the results by typing a cutoff value in the text box under each column name. When the filter rows are less than 100, we can automatically view these plots in the data viewer. The plots are connected. Click to select a data point in one plot automatically. Highlight the same data in the other plot. To view the pathway, go back to the pathway report. Click on the gene set link in the first column. The pathway is colored by genes from our filtered features list. We can hover over this part of the pathway. In bold, we see the genes from our filter feature list that are implicated in this part of the pathway. We can also see additional parts of the pathway that may be important. And go ahead and save this image to our machine as well. Let's go back to our analysis pipeline. To summarize, we start our analysis by reviewing methylation calling quality in QC report and exploring source of variations in PCA. We then use DSS to detect differentially methylated regions across the two sample groups, filtered the DMRs by test statistics, annotate the regions with gene names, and identify potential pathways for biological insights. This should give you an end-to-end overview of 5 base data analysis in the Illumina Connected Multiomics.